हेलो फ्रेंड्स, अ वार्म वेलकम टू दिस चैनल साइंस प्लेस। आज हम बात करने वाले हैं हिस्ट्री एंड इंट्रोडक्शन ऑफ माइक्रोबायोलॉजी की। सो लेट्स बिगिन द चैनल टॉपिक। इन दिस टॉपिक we are going to study about discovery of microorganisms, theory of spontaneous generation, how that was disagreed, and germ theory of disease. So before starting the topic, uh, do we think anytime what is a microorganism? Microorganism is any living body that cannot be seen with our naked eye or unaided eye. Means without any lens, without any microscope, if we are not able to see any organism, that will categorize as microorganism. So if microorganisms are present surrounding to us, yes, in the air, in the water, in the soil, these organisms are present. Any water body like river, pond, lake, that is having a lot of microorganisms. And like soil is like hub of microbes. So and even our body, it is having a lot of number of microorganisms. So microorganisms can be categorized as bacteria, virus, fungus, algae, protozoa, and even the surrounding air of us is having microorganisms. So abhi to last time we seen that covid, the virus that we cannot see with our eyes, but that was transmitted by the close intimacy of contact of infected person. The probable source of contamination infection was the air, because one person when he or she was releasing the covid virus, that other was inhaling and was getting infected. So now because of very high sophisticated instruments, we are able to know that yes, microorganisms are present in our surrounding. But how they were discovered first? So discovery of microorganism was initiated with the application of microscope to see living organism, living thing. The first use of microscope to see the living thing was by Robert Hooke. Robert Hooke, he was a scientist, and he first time he seen the cork, a thin slice of cork, and he was able to see very small boxes in that cork. He used a binocular microscope that was having two lenses, and when he seen the slice of that cork, he was surprised to see that there were small boxes, and he remind the bare rooms of monks at that time, so he termed the coin cell for these empty boxes. And later on, cell is another thing that is not the microorganism, but yes, the to see the living thing started with the application of microscope by Robert Hooke. And later on he seen a lot of plant cells, fungal structures, and molds, and he compiled all his results in his book, that is Micrographia, that was published in 1665. And he was able to view and build this microscope because he was a scientist, and he was used to make telescope basically. And telescope mein he was able to observe the rotation of Mars and Jupiter. So by using his physics knowledge, he made this microscope and seen the cell first time and coined the term cell. After that, Robert Hooke, he published Micrographia. In parallel to Robert Hooke, there was a child, can say child, or some young person of 22 years of age. He was a draper basically, and his father was a basketman. So he was a poor guy. He opened a shop of clothing draping in Dutch, and he was a having formal schooling only, but he was earlier from opening this shop, he worked in a bookshop, and there he seen the microscope at that time. So he was very keen to see the microscope, and later on, because of his interest, and because he used to apply lens, he used the lens to see the quality of fibers of that cloths. He used to see the different kind of things surrounding him by using microscope. So he used to see, water around around him, like pond water, and he used to see blood cells also, even cells he seen in animals and human also, and lot of things he seen. But because he was not a scientific person, so he compiled his results and sent to uh, a very prestigious Royal Society. A lot of letters he wrote them, but many times they get refused. But ultimately, uh, he described the picture of some microorganisms. Here you can see different shapes of bacteria are there by using this simple microscope. The microscope is made up of only single lens. So you can see here is the single lens, and on the tip of this needle, he used to put the sample. So by using this microscope, he draw this diagram and sent to the Royal Society. And in 1676, his letter was accepted, and he was, rewarded for this discovery first time. But at that time, he termed the coin he gave the term animalcules for those small creatures, not the bacteria or fungus or any other thing. Because in Dutch, that means he originally referred these microbes of daikens. That means in Dutch, that is the meaning of small animals. So by using a simple microscope made by Antonie von Leeuwenhoek, he could presented this diagram with different shapes of bacteria, and this was appreciated by Royal Society of uh, Royal Society. So, because of this discovery, later on, Anthony Phillips von Leeuwenhoek was rewarded as father of microbiology. But after his discovery, the microbiology cannot grow enough because he denied to share his own microscopes to other scientists. But science goes on like that only. No one can hide the things. So in after that, there was a medicine professor at the Berlin University, who described many new types of cells, protista, diatoms, and he was very keen to see sedimentary rocks. And he shown that sedimentary rocks are these who looks like chalk. They are composed of many microscopic cells. And later on, he seen some bacteria that was rod shaped, and he coined the term bacteria in 1838. So this is a brief of how bacteria was discovered by you know uh, efforts of many scientists, which I'm not going to describe here, but basically two scientists, they contributed a lot for the discovery of bacteria. After that, in parallel to this, there was a discussion about the spontaneous generation. A lot of scientists believe at that time that life originated from non-living material, non-living matter. Even the Aristotle, he was a very famous person, but he believed that the life is made up of fire, earth, air, and water, using four elements. And so people seen that grains are having rodents are arose from moist grains, and beetles from the dust, and worms and frogs can be rise from mud. But these theories, and many scientists even believe that rotten meat can give rise to worms. But there were very big scientists who not appreciated well in the microbiology, Francesco Redi. he shown in his experiment that living things not not came from non-living material. How he designed the experiment? He make a experiment of 3 bottles. You can see here in this, one bottle is having, all the 3 bottles are having piece of meat, and one is open, one is sealed with cork, and other is sealed with the cloth only, gauze cloth only. So he concluded from this experiment, he kept this set of experiment for a short duration, and seen that the open meat piece is having uh, these worms appeared due to, because maggots, these are the flies which produce eggs on the meat, and they ultimately produce the maggots. But in this second bottle that was sealed completely, there is nothing produced. In another bottle that was not, that was sealed, but air was passing from that bottle, the maggots are not at the piece of meat, but they are at the orifice of the bottle, because here the smell can be passed out from this meat source, and here maggots are produced. But this his observation was not appreciated much in the scientific community, and people continued the experiments further, and finally the theory of spontaneous generation was defeated by the postures of experiments produced by Louis Pasteur and John Tyndall. What happened to, how they disproved this theory? That Louis Pasteur, he was a very famous professor at that time of chemistry, and he used to examine different crystals of chemicals using lens, microscopes, microscope and he observed that during the production of alcohol, different type of tartaric acid crystals are produced, and give the concept of optical isomers. Optical isomers are the particles that can rotate the light either left or right side, levorotatory or dextrorotatory. That is not part of this topic, that's why I'm not going to cover this. But he was a very famous chemistry professor. So what happens, once a very businessman came to Louis Pasteur, he was very sad because his not production was not going well. Why? Because he used to make alcohol by using beet juice, but his batches was not producing alcohol, but the end product was a sour milk smelling compound, that he don't know about that. So he came to Louis Pasteur ki, sir, this is my problem, how to solve this? I'm very fed up with this. I'm not able to produce alcohol, heavy loss in business, this and that. Oh. So Louis Pasteur decided to help that person. And what he did? He seen that, okay, he seen the product of the his um, vats under the microscope. He seen the broth under the microscope in both the batches. In one batch there was production of alcohol, in other batch there was production of this bad smell compound, the sour smell compound. So he, under the microscope, he not seen the crystals, but he seen different kind of cells. And what kind of cells? The alcohol in the pot, in the vat that was producing alcohol, was full of yeast. Yeast is a kind of fungi. And in the vat that was producing the raw that sour product, bad ordered, that was full of rod shaped bacteria, you know. So there was a difference. He seen the difference, and later on he performed some experiments and concluded that this was the thing that was producing alcohol, and this rod shaped bacteria were producing the some acid, lactic acid something. So, and later on he also gave the term fermentation for this procedure, production of alcohol. That is a separate story. So by using his microscopic study, he solved the problem of that businessman. When you are your what will having is, then you will able to produce alcohol, otherwise not. Okay, so after this interesting finding, he also performed a he was a, because of famous professor in 1859 there was a competition organized by French Academy of Science, and in that competition, the topic was to try by well performed experiment to throw new light on the question of spontaneous generation. So Louis Pasteur appeared in that competition, and he performed this very famous swan neck experiment. This flask is called as this flask is called as swan neck flask because of this shape of swan. And how this he performed this experiment, he first put a he took a normal flask. This is the normal flask, and he filled this normal flask with the non-sterile liquid solution, liquid media, to grow the bacteria or whatever it is. This is simple non-sterile liquid poured in the flask, and in next step he used the flame to, here you can see he used the flame to melt the to shape the flask like a swan. And in next step he he used the heat to sterile this liquid, because in his fermentation wala experiment he was able to know that living things can be dead by using heat. So he used this that concept here, and by sterilization by heat, he produced this kind of swan necks flask with sterile medium. And he kept this media liquid after a time that get cold, and he kept this flask for a very long time. But because of this particular shape, there was no pass of microorganism from this way to the liquid. That's why after long time also that liquid remained sterile. But when he dipped the flask, and you can see here that liquid passed through this tube, and when touched and came back to the liquid again, the original place again, here is the contamination, the liquid get putrefied. So by using this simple and beautiful experiment, he concluded that because microorganisms get trapped here for a longer duration, this liquid was sterile. But when this came back after touching this dust and the portion that was captured from air, the liquid get contaminated. And he concluded that uh, there is no theory like theory of spontaneous generation. He disapproved and disapproved the theory of spontaneous generation. So in this lecture I'm going to stop here. Thank you for watching this video, and if you are liking this, please press the like button and subscribe this channel. And you're welcome, suggestions are strongly welcome. I will take your suggestion comments to improve my video. So thank you so much. Next part will be after some time. Thank you. Bye.